Thanks for joining us on another exciting journey into the world of science, right here on the Sand Science Experience. If you enjoy this kind of content, hit like and subscribe. It's the best way to support the channel and keep these episodes coming. Today, we're diving deep into a really fascinating recent scientific discovery, a potential treatment for a truly devastating and currently untreatable mitochondrial disease called Leigh syndrome, or LS.
That's right. Leigh syndrome is a severe genetic disorder that, it messes with how the mitochondria, the powerhouses of our cells function. This leads to a whole cascade of problems, particularly in young children, causing psychomotor regression, those heartbreaking losses of skills they've already learned, and terrifying metabolic crises. Sadly, it often results in early death.
It's such a cruel disease, and what makes it even harder to tackle is that over 100 different genes can be involved. We're talking about genes crucial for different parts of the energy-making process, like the gene for complex 5, MTATP6, a gene involved in assembling complex 4 known as SURF1, and even genes for complex 1, such as Ndufs4.
And as you can imagine, with so many potential genetic culprits, finding effective treatments is, it's a massive challenge. One of the big hurdles has been the lack of good models to study the disease and test potential therapies. The Ndufs4 knockout mouse has been a go-to model, particularly because it mimics the brainstem pathology seen in many patients.
But even that model has its limitations, right? I mean, it's a mouse, not a human, and sometimes what works in mice doesn't.
Well, it doesn't translate. That's why this new research we're looking at today is so exciting. It takes a completely innovative approach by using patient-derived induced pluripotent stem cells, or iPSCs. Okay, let's unpack this. These are basically stem cells created from a patient's own cells, yeah? And the amazing thing is they can be turned into any other cell type in the body, including the ones affected by Leigh syndrome.
Exactly. This allows researchers to create disease-relevant cellular models right there in a dish, models that actually carry the patient's specific genetic defect. So instead of a generic model, you have something that can show patient-specific issues. So the mission for this deep dive is to explore how these researchers use these patient-derived iPSCs to do something really clever. They screened thousands of existing drugs that are already approved for other conditions, to see if any of them could potentially help with Leigh syndrome. And guess what? They found a surprising potential treatment. It's like finding a new use for an existing tool in your toolbox.
It's a fantastic example of drug repurposing, which can, significantly speed up the process of finding therapies for diseases like Leigh syndrome, where time is so critical.
All right, so let's get into the nitty-gritty of how they did this massive drug repurposing screen. What were the researchers actually looking for in these iPSC-derived neural progenitor cells, or NPCs?
What's fascinating here is that the researchers had previously observed a specific abnormality in LS NPCs with these MTATP6 defects. Their mitochondrial membrane potential, or MMP, was hyperpolarized. Think of MMP as the electrical charge across the inner membrane of the mitochondria, crucial for energy production. In these disease cells, that charge was just too high. It's like a battery that's overcharged, it's not working efficiently, and can even become damaged. Sildenafil seems to help dial that charge back to the optimal level.
So this hyperpolarization became their target. They wanted to find drugs that could bring that MMP back down to a normal level. Yeah. How did they actually screen thousands of drugs for this? It sounds like a huge undertaking.
It was a very sophisticated process called high-throughput screening. They used a library of 5,632 compounds.
Wow.
That are already approved for use in humans, so their safety profiles are generally known. They then used high-content microscopy to automatically look at the MMP in the LS NPCs after treating them with each of these compounds.
And how did they measure this MMP? Was there some sort of tiny voltmeter involved?
Not quite. They used a fluorescent dye called TMRM. This dye is sensitive to the mitochondrial membrane potential. The more polarized the membrane, the more the dye accumulates and, the brighter it glows. So by measuring the intensity of this fluorescence under the microscope, they could tell if a drug was affecting the MMP.
Okay, that makes sense. So they basically had thousands of tiny experiments running at once, looking for changes in this fluorescent signal.
Yeah.
And out of all those compounds, they found some promising leads, right?
Exactly. They identified a class of drugs called phosphodiesterase type 5 inhibitors, or PDE5I for short, as being particularly good at normalizing this MMP defect in the LS cells. Interestingly, they had even seen hints of this with another PDE5I called avanafil in some earlier, smaller studies.
PDE5 inhibitors.
Oh,
that sounds like a mouthful. What exactly are they inhibiting, and why would that be helpful here?
That's a great question. PDE5 inhibitors block the action of an enzyme called phosphodiesterase type 5. This enzyme normally breaks down a molecule called cyclic GMP, or cGMP. By inhibiting PDE5, these drugs allow cGMP levels to remain higher, and as we'll see later, cGMP plays a role in some important cellular processes.
Ah, okay.
And yes, they had seen hints of this class of drugs being beneficial in earlier research.
Okay, that's helpful context. Now, PDE5 inhibitors, that rings a bell. Isn't sildenafil, often known by the brand name Viagra, in that same class of drugs?
You're absolutely right, and this is where it gets really interesting. While they found a couple of PDE5 inhibitors that look promising, they prioritize sildenafil. The reason? Its well-established safety profile, not just in adults, but also in infants and children who use it for conditions like pulmonary hypertension.
So, a drug with a known safety record in vulnerable populations jumps to the front of the line. That's smart thinking. What did they find when they tested sildenafil more closely on the LS cells?
They found that sildenafil was able to depolarize that overly hyperpolarized MMP in the LS iPSCs in a dose-dependent manner, meaning the more sildenafil they used, up to a point, the more the MMP normalized. Importantly, this happened without causing significant cell death, and the concentration at which it had a 50% effect, the IC50, was around 3 micromolar.
And crucially, did it have the same effect on healthy cells? You wouldn't want a drug that messed with mitochondrial function in everyone, right?
Precisely. And that's another key finding. Sildenafil did not significantly affect the MMP in control NPCs, which is a really encouraging sign that its effects might be specific to the disease cells.
So sildenafil seemed to be fixing this electrical imbalance in the mitochondria of the LS cells. But what about the actual energy production? I mean, did it help with that?
That's the next logical step they investigated. They looked at the activity of complex 5, that ATP-producing machinery, and found that sildenafil treatment in the LS NPCs made its activity more similar to what they saw in the control cells.
That's a big deal, right? If the energy-producing machinery starts working more normally.
Absolutely. And they pushed the cells even further by growing them in a glucose-free medium that contained galactose. This forces the cells to rely heavily on mitochondrial respiration for their energy. What they found was that sildenafil actually increased the intracellular ATP concentration in the LS NPCs under these stressful conditions.
So not only was the electrical charge normalizing, but the cells were actually producing more energy when they really needed it.
That's fantastic. They also looked at the physical structure of the mitochondria, right? The cristae.
Yes, because MT-ATP6 defects are known to sometimes disrupt the inner folds of the mitochondria called cristae. They used a sophisticated type of microscopy called STED microscopy to get a really detailed look at the distribution of a protein called MIC60, which helps regulate the junctions of these cristae. Using machine learning to analyze thousands of images, they found that sildenafil partially reversed the abnormal distribution of MIC60 in the LS NPCs, suggesting it was helping to restore a more normal mitochondrial structure.
Wow. So to recap, sildenafil was normalizing the electrical potential, improving energy production under stress, and even helping with the physical organization of the mitochondria in these LS cells. That's a pretty compelling set of initial findings.
It really is.
But these findings naturally led us to ask how sildenafil was achieving these improvements at a molecular level. That's where these really powerful omics techniques came into play.
Omics. It's like looking at the whole picture at once, right?
Yeah.
Genes, proteins, metabolites, all of it.
Exactly. So instead of looking at one gene or one protein at a time, omics lets researchers take this massive snapshot of everything happening inside the cell simultaneously, giving them a much more complete picture of the disease and how a drug might be changing it. They use transcriptomics to look at all the gene activity, proteomics to see which proteins were present and in what amounts, and metabolomics to analyze all the small molecules, the metabolites, within the cells. They did this on both the LS NPCs and the healthy control NPCs, both with and without sildenafil treatment. This allowed them to identify what they called the disease signature, what's different in the LS cells, and the sildenafil signature, what changes when they treat the LS cells with the drug.
And what did these signatures reveal about what was going wrong in the LS cells and how sildenafil was helping?
The disease signature showed a lot of dysregulation in processes related to the nervous system and the development of axons, those long projections of nerve cells. But the sildenafil signature showed a reversal of these processes. For instance, a gene important for neurodevelopment called DBX1 was down in the LS cells, but went up after sildenafil treatment. Similarly, a protein called BAG4, which helps protect cells from ATP depletion, was low in LS cells and increased with sildenafil. Interestingly, these changes seem to be specific to the LS cells, not really happening in the healthy control cells treated with sildenafil.
Wait.
So what we're really seeing here is that sildenafil isn't just tweaking one thing, it's hitting multiple pathways that are going wrong in these Leigh syndrome cells, bringing a lot of these key biological processes back towards normal. That's a pretty profound insight into how this drug might be working.
That specificity is really important. What about the metabolites? Did sildenafil have an impact there?
Yes, metabolomics showed that key metabolites like creatine and cysteine were abnormal in the LS cells, but sildenafil helped to normalize their levels. It also significantly affected the glutathione metabolism pathway, which is important for dealing with oxidative stress, and it even normalized the ratio of NAD plus to NADH, which is crucial for cellular energy metabolism.
Wow, so sildenafil seems to be hitting multiple targets at once, helping to correct a wide range of problems in the LS cells. And then they integrated all this omics data together, right, to see the bigger picture.
They did. By looking at the whole interconnected network of genes, proteins, and metabolites, they found that sildenafil was able to reverse about 33.5%, about a third of the dysregulated molecules in the LS cells, particularly those involved in mitochondrial respiration and the development of nerve cell structure, like morphogenesis and axon development. One molecule that really stood out in this integrated analysis was cGMP-dependent protein kinase 1, or PRKG1.
Ah, PRKG1.
Yeah, this is a known target of PDE5 inhibitors like sildenafil, so it starts to paint a clearer picture of the drug's mechanism.
So this PRKG1 might be a key player in how sildenafil is exerting its beneficial effects. That's a crucial piece of the puzzle. But these were still just cells in a dish, right? They wanted to see what happened in a more complex model.
Exactly. So the researchers moved on to using 3D brain organoids. These are tiny, lab-grown structures that mimic some of the complexity of the human brain, allowing them to study neurodevelopmental defects in a more realistic context.
Brain organoids, okay. And what did they see in the LS brain organoids? Were the same kinds of problems showing up as in the simpler cell models?
Yes, consistent with other studies, they found that the MTATP6 mutations impaired the formation of neurogenic zones, the areas where new neurons are born. They also saw an altered ratio of early neurons to neural progenitor cells, suggesting problems with how these progenitor cells were developing.
So the building blocks of the brain seem to be affected. What happened when they treated these brain organoids with sildenafil?
They tried two different treatment approaches: a short 24-hour acute treatment and a longer 45-day chronic treatment. With the acute treatment, transcriptomics showed that sildenafil affected pathways related to embryonic development and WNT signaling, which are important for early brain development. It even corrected the levels of a specific gene called WDR45B, which is linked to neurodevelopmental disorders.
And for the longer treatment, they used that really detailed single nucleus RNA sequencing to look at individual cells within the organoids, right? Like cell by cell.
That's right. This allowed them to see which specific cell populations were most affected by the MTATP6 mutations and by the chronic sildenafil treatment. They identified different types of cells, including radioglia, progenitors, and various types of neurons. The MTATP6 mutations seem to impair the commitment of cells to become neurons, affecting the populations of radioglia, progenitors, and FOXG1 positive neurons. They also saw a downregulation of genes like NLGN1 and PRKG1 in progenitor cells and STMN2 in neuronal cells.
And what was the impact of the long-term sildenafil treatment on these specific cell populations?
Interestingly, the chronic sildenafil treatment primarily affected those same populations: radioglia, progenitors, and FOXG1 positive neurons. It led to the upregulation of a gene called RGS6, which is involved in neuronal outgrowth. Don't seem to be adding any additional stress to the organoids, which is good news. The glycolytic stress signature wasn't altered.
So in these more complex brain-like structures, sildenafil was still having targeted effects on the cell populations that were most disrupted by the Leigh syndrome mutations, particularly those involved in early brain development.
Exactly. And then they moved on to look at functional improvements, focusing on calcium signaling and neurite outgrowth, two things that are often affected in Leigh syndrome.
We touched on calcium briefly earlier. What did they find in the brain organoids under metabolic stress? How did they react?
They found that slices of the LS brain organoids showed a much more pronounced and premature increase in calcium levels when they were subjected to metabolic stress, essentially depriving them of energy. This suggests that the disease cells are much more vulnerable to energy shortages. However, when they pre-treated the LS organoid slices with sildenafil, this abnormal calcium response was significantly reduced.
Wow.
So Sildenafil seemed to be helping the cells better handle metabolic stress, potentially preventing those severe decompensations that are so dangerous in Leigh syndrome.
And this was supported by some computational modeling, right? Like computer simulations.
Yes, the computer model suggested that the ATP synthase defects in LS could lead to this elevated calcium response, and that sildenafil's effects on MMP, NADH, and ATP levels could be modulating this. What was particularly interesting was that sildenafil even normalized MMP and NPCs with SCRF1 mutations, where the MMP was actually depolarized, not hyperpolarized.
Oh, right. So it worked even when the starting problem was different.
Exactly. This hinted that sildenafil's benefits might go beyond just directly uncoupling the mitochondria, suggesting a broader mechanism at play. That's a really key insight.
That's a really important point. It suggests a broader mechanism of action. And then there was the impact on neurite outgrowth, the growth of those crucial neuronal connections.
They found that sildenafil promoted neurite outgrowth in neurons with mutations in MTATP6, SURF1, and even NDUFS4.
So across different genetic causes.
Yes, but it didn't do this in control neurons. This suggests that sildenafil might be specifically helping to rescue the impaired neuronal development in Leigh syndrome.
And this all seemed to tie back to that PRKG1 protein we talked about earlier, right? That kinase.
Absolutely. They found that PRKG1 levels were lower in the LS models. When they inhibited PRKG1, it blunted sildenafil's effect on MMP. And when they actually knocked down PRKG1 in healthy neurons, it mimicked the neurite growth defects seen in the LS neurons. This strongly suggests that sildenafil's beneficial effects are at least partly mediated by its action on PRKG1.
So sildenafil seems to be working through this PRKG1 pathway to improve mitochondrial function, calcium homeostasis, and neuronal development in these lab-grown models of Leigh syndrome. That's a really exciting chain of evidence.
Yeah.
So we've seen these really promising effects in the lab using these patient-derived cells. But the big question is, do these findings hold up in a more complex living system? Let's see what happened when they moved to the mouse model.
It is, but first, for a drug to work in patients with Leigh syndrome, it needs to be able to reach the brain. So the researchers investigated whether sildenafil could cross the blood-brain barrier.
And what did they find there? It can be a pretty tough barrier to get through, can't it?
Using a human iPSC-derived model of the blood-brain barrier, these brain capillary endothelial cells, they found that sildenafil and its orally administered form, sildenafil citrate, could indeed cross the barrier. Its permeability was similar in both control and LS models, suggesting it should be able to reach the brain in patients.
Okay, that's another crucial piece of the puzzle in place. And then they took it a step further and looked at the effects in a living organism, using that Ndufs4 knockout mouse model of Leigh syndrome.
Yes, this in vivo study yielded some very encouraging results. They found that sildenafil treatment significantly extended the lifespan of these mice. It also helped to reduce their neurological decline, alleviating muscle weakness and problems with coordination, ataxia.
That's huge. Extending lifespan and improving neurological function in an animal model of such a severe disease is a major step forward.
Absolutely. They also saw improvements in the mice's energy expenditure, oxygen consumption, and carbon dioxide production, suggesting an improvement in their overall metabolic health. Even their heart function, which is often affected in these mice, they saw amelioration of cardiac bradyarrhythmia and dysfunction.
And at the cellular level in the mice, were they seeing similar benefits to what they observed in the human cells, in the brainstem and cerebellum?
Yes. They found a reduction in signs of apoptosis, or cell death, using cleaved caspase-3 staining and neuroinflammation, looking at ABO1 staining in the brainstem and cerebellum of the treated mice. They also saw preservation of Purkinje cells, a type of neuron particularly vulnerable in Leigh syndrome, and a rescue of that PRKG1 expression we discussed.
So the benefits seen in the human cellular models really seem to translate to this animal model of the disease. That's incredibly promising.
It is. And this strong preclinical evidence led the researchers to take a bold step and explore compassionate treatment in actual patients with Leigh syndrome.
Right. This is where it gets really personal and impactful. They treated 6 patients with Leigh syndrome, all with different mutations in that MTATP6 gene, on an off-label basis. What did they observe in these patients?
Firstly, they confirmed that sildenafil did reach therapeutic concentrations in the patient's blood, around 1 micromolar, which is close to the effective concentration they saw in the lab. Importantly, the drug was generally well tolerated by these very sick children.
That's good to hear. And what about the clinical outcomes? Did they see any improvements in these patients who had previously had no effective treatment options?
In all 6 patients, they observed clear improvements in various clinical parameters, particularly in motor function, muscle strength, and their resistance to those dangerous metabolic crises. For example, patient 5, who used to have metabolic crises almost monthly, didn't have any after starting sildenafil.
Wow.
Some patients also showed mild cognitive improvements. It was even noted that when patient 3 had to stop sildenafil, their symptoms worsened again.
That's powerful anecdotal evidence. And they also use those standardized scales to track the progression of the disease, right? The NPM, DSM, MDS score.
Yes, they used the Newcastle Mitochondrial Disease Adult Scale and the Newcastle Pediatric Mitochondrial Disease Scale. Typically, these scores increase over time as the disease progresses, but remarkably, in these 6 sildenafil-treated patients, the scores either decreased or showed a much slower rate of increase compared to historical data from untreated patients with similar mutations.
That's a really compelling finding, suggesting that sildenafil might actually be modifying the course of this devastating disease. But they are careful to point out that these are preliminary findings from a small group of patients, right?
Absolutely. While these compassionate use results are very encouraging, the researchers emphasize the critical need for a formal, randomized, controlled clinical trial to definitively evaluate the safety and efficacy of sildenafil in a larger group of patients with Leigh syndrome.
Of course. These are incredibly encouraging stories. But obviously, with such a small number of patients and no control group, we need to be cautious about drawing definitive conclusions.
Yeah.
A full trial is the crucial next step.
Exactly. That's essential to really prove whether sildenafil is effective in this patient population.
So to summarize this deep dive, we've seen how researchers used an innovative iPSC-based approach to screen thousands of existing drugs and identified sildenafil, a readily available medication, as a promising potential treatment for the severe mitochondrial disease Leigh syndrome.
That's right. We've seen how sildenafil had multiple beneficial effects in preclinical models, normalizing mitochondrial function, improving neuronal development, calcium homeostasis, and neurite outgrowth, and even extending lifespan in a mouse model. And we've also seen those initial encouraging signs of clinical improvement in a small group of patients treated with sildenafil.
It really highlights the power of these patient-derived cellular models in uncovering unexpected therapeutic candidates. It makes you wonder, how many other existing drugs might have untapped potential for treating rare and challenging diseases where few options currently exist?
It certainly does raise that important question. This research offers, well, a beacon of hope for families affected by Leigh syndrome and underscores the potential of repurposing existing drugs to accelerate treatments for a wide range of other conditions.
Thanks for listening. And as always, don't forget to like the video and subscribe if you haven't already.